Okay, so hello everyone, and welcome to this webinar. It's my great pleasure today to introduce to you Dr. David Dobnik for today's webinar on transforming biopharma, the impact of digital PCR and quantification in biopharma research and manufacturing. So David has been working in the field of molecular biology since 2007, with a focus on the development of new nucleic acid detection and quantification approaches. In the past few years, David has become an expert in digital PCR assay development, and particularly in high multiplex assay development. He is currently a senior researcher at the National Institute of Biology in Slovenia and also the chief scientific officer at NIBA Labs, which is an expert lab in digital PCR assay development for viral vector characterization to address the analytical needs of gene therapy products. A few housekeeping rules before we start this webinar. So if you have any questions, please write them down in the questions tab, and we'll be answering questions at the end of the webinar for about 5 to 10 minutes. Throughout the webinar, you'll have poll questions that will be popping up on your screen. If you can, please answer these. And the overall result of the poll will be shared at the end of the webinar. So now, without further ado, I leave the floor to David. Thank you, David.
Thank you, Alice, for a nice introduction. And welcome everybody to this presentation on digital PCR in general and how it impacted the biopharma research and manufacturing field. So just a quick disclaimer that these are my opinions and the presentation was supported from Stila. In today's talk, I would like to make some introduction on just present myself, the background. Then we will kind of explore the evolution of PCR and how it came to digital PCR, as also I view it and as I view it in the context of biopharma. And the last part, I would like to focus also on the impact in terms of using multiplex capabilities of digital PCR. And I will also present a case study of multiplex assay that can be suited for the gene therapy characterization needs. And we will conclude then with some discussion and Q&A session in the end. So I come from a molecular biology, biotechnology background, and I did a lot of development on the new assays, or previously at least on the GMO quantification, plant pathogen, also non-PCR based methods. I did a lot of functional genomics of potato PVY systems, so completely out of biopharma, and some small RNA studies as well. Currently, I'm mostly involved in quantification, characterization, and development support in the field of viral vectors for gene therapy. I still had a lab for official detection of GMOs at NIB. And let's say I'm also involved in completely other field, that is the contamination of liquids by use of plasma and cavitation technology. Because Alice mentioned also NIBA Labs, I need to tell you this, that I'm in gene therapy since 2016, and I'm also co-founder of NIBA Labs, so not just chief scientific officer. And this is a spin-out from National Institute of Biology that kind of built upon the knowledge that we had at NIB in terms of molecular biology and virology, where we started working on supporting other companies in the field of gene therapy, such as Avexis. And we actually transferred this knowledge to the company, so NIBA Labs, and now we have a mission with NIBA Labs to improve the speed and efficiency of process development in a segment of cell and gene therapies by providing an expert support in development and analytics. We have labs that are located at Novartis Industrial Campus in Slovenia. Okay, now we can go to the topic of evolution of PCR. So PCR is it's a very useful technique already from the start. And you probably might remember or still perform classical PCR reactions as well, where the reactions are more or less qualitative. You can do at least some extent of quantification. It's more like a semi-quantification based on the intensities of the bands. But as said, it's more or less for qualitative purposes, just to kind of detect the signal and see, let's say, the size of the bands. Then kind of the evolution in terms of PCR came with real-time PCR, where we could follow the fluorescence of targets that were being amplified in real time through the fluorescence. However, this was still a relative quantification. You require a standard curve, some certified reference material to which you then compare the signal from your sample. But in between these times, so even before appearance of real-time PCR, there was already, let's say, initial spark ignited for the digital PCR with the concept of limiting dilutions that was introduced already in 1992. So the limit of dilution is actually dilution of the sample at the point where the signal in the PCR starts disappearing, meaning that if you test this sample in several reactions, you would get some positive or negative responses of those reactions in the end. And if you then use the Poisson statistics based on the proportion of these positive or negative reactions, you can already get, let's say, a bit more absolute quantitative way of the result. As said, this concept came between PCR and real-time PCR, but can be actually employed for both of these techniques. But what we lack with these techniques is that there's really not a lot of replicates, so not a lot of reactions that can be used. And there actually the real digital PCR came in with the development of the microfluidics that enabled actually partitioning of the initial reaction mixtures into several hundreds, thousands, or even millions. Digital PCR is in fact an endpoint PCR reaction, and you get this kind of digitalization of the signal by positive or negative reactions, so 1, 0. It enables really absolute quantification of targets, and that's of course kind of enabled with the many partitions that we have. In, let's say, basics, it's actually almost the same as real-time PCR, because you need primers, probes, some master mix and your sample for the reaction. But then the difference is actually in the splitting of this reaction into, as mentioned before, hundreds, thousands, or even millions of partitions. After that, once you have the partitions, you amplify the targets through PCR cycling. And in the end, you need to score each of these partitions, again, if they are positive or negative. When you have these raw results, usually the software calculates the concentration that is based on the Poisson statistics. So if we now perhaps go and check the history of digital PCR at the beginning, and also then our activities connected to digital PCR, as mentioned, 1992, the concept of the digital PCR was conceived, And then the first paper really introducing the word digital PCR was published in 1999. What followed was a period, perhaps, of developments, and you can see that the first commercial digital PCR system that was based on microfluidic chips became available in 2006-2007. First commercial system that was based on the water-oil emulsion, or let's say droplet digital PCR system, was launched in 2011 by QuantaLife, which was actually very soon taken up by the Bio-Rad, which introduced its own QX100 system in 2012. And at that time, at the Institute of NIB, we got actually the first dPCR system that was installed in Europe. And after that, we've used it for very different activities. However, the first one that we also published was on the suitability of its use for quantification of GMOs in food and feed samples. We continued with several studies on plant material quantification, like viruses, also on the one-step approaches, also bacteria quantification. However, our, let's say, main topic at that time was already to be, let's say, somewhere where they did not think about using PCR, and that's for higher multiplexing. So at the beginning, this system was a double fluorescence channel system. However, we went further and developed an assay that worked in higher multiplexing level at that time. In 2016, it was the first digital PCR system with more than 2 fluorescence channels that was launched. That was Stila's NICA Crystal Digital PCR. And roughly at the same time, we then also went further with our multiplex development and we developed a multiplex assay that was able to quantify 4 targets separately in one reaction. And actually, all of this expertise that we gained in those years from the beginning was recognized in 2016 by Avexis. And this was our beginning of collaboration with them and actually how we entered into the field of gene therapy. As I mentioned before, we had knowledge on the virology. It was plant-based virology, but essentially it was viral viruses that were used and quantified with digital PCR, and we had a lot of knowledge on the molecular biology as well. So we developed their titer assay that they use now for the release, transferred it to their labs, and we supported them as well with some other characterization methods in their process development. After that, we actually started presenting our work on digital PCR in connection with gene therapy at the conferences. And we started collaboration also with other companies performing actually kind of very similar activities as with Avexis. We also initiated some project on lentiviruses in 2018. But perhaps if we focus on our beginnings going into the gene therapy, I will, just for the listeners that are not from the gene therapy, mention the gene therapy as such. So the idea is to deliver some gene construct to the cells of the patients that need the gene replaced or some other protein being expressed. And viral vectors are actually delivering this payload to the cells, and viral vectors need to be well characterized so that we really know what they deliver. So there are different ways how viruses can work in the gene therapy. It can be like this, so they are being directly administered to the patient's body, and they do not integrate in the genome, but of course they do trigger some expression of the protein, for instance. With integrating viruses, they can be used for, let's say, ex vivo treatment. This is an example, let's say, of CAR T cells, where you modify patient cells outside of their body, do some amplification, and then put them in the patient's body where they can fight the disease. Or let's say viruses can be modified in a way so that they target specific cells, let's say tumor cells, where they cause their disintegration. So to characterize all those viruses that can be used as drugs, there's of course a lot of different methods that need to be used. And as you can see, digital PCR as well as qPCR do play some role in characterization as well. So focused, of course, on the viral genomes and the impurities that might be present. And what is actually the power of digital PCR for quantification of any nucleic acid, not just viral vectors? Definitely the first one is that you can determine absolute copy numbers without any need of standard curve. With many applications, it's very difficult to get material that would be characterized, so certified, that you could really use for the standard curve. And here, digital PCR plays quite a big role. It also offers accurate and precise quantification, even at very low copy numbers that are present in a high background of other targets. Moreover, the results are very repeatable, and the digital PCR is much more tolerant to inhibitors than, let's say, the qPCR is. It's not, let's say, completely tolerant. There still can be some inhibitors that do affect digital PCR reaction, but overall, very better tolerance than qPCR. And what's also the good thing is that if you already have the assays that worked in qPCR, the transferability to digital PCR can be rather simple. Of course, there are some things that you need to check to be sure, but let's say majority of assays can be transferred to digital PCR quite easily. So what actually we have seen in the history of our involvement in the digital PCR was that actually it was very quickly adopted in oncology and a bit slower in gene therapy. So after initial adoption of digital PCR, as mentioned, it was not really adopted well in really diagnostic community. However, perhaps in cancer diagnostics, yes, but it was more used for the research. In terms of gene therapy, first dPCR method was published already in 2014, and then, let's say, few individual papers per year kind of followed, but not really a lot. Now, why this slow adoption rate in gene therapy? One of the reasons could be a bit more strict regulatory systems for products that are already in clinical trials. And let's say companies were a bit reluctant to kind of change the qPCR technology, which was already in their files with the regulatory agencies. And they just kind of kept digital PCR on site. And actually, when I attended my first conference on gene therapy in 2017, I was presenting our work on viral vector genome titer determination. And I can say that digital PCR at that time was considered still quite exotic. So I was presenting really on the basics, how digital PCR actually works, what it can be used for. And then in the following years, I have seen that more and more presentations in the gene therapy field conferences did include the data generated by digital PCR. And today, definitely digital PCR is considered already as a gold standard. For viral vector titer quantification, one example from our paper is here. Let's say why people decide for digital PCR is because of the really lower variability in the results that you can get in terms of qPCR. And with this adoption now, there are projections that even propose a 10 times increase in digital PCR market size in the next decade. So we are, let's say, starting this exponential growth in the adoption of digital PCR. But we can now go, let's say, into more specifics of digital PCR as such. So we are talking about simplex, duplex, even multiplex digital PCR. Just to put us all on the same level, when speaking about simplex, it's just one target per reaction that is being amplified and detected and quantified. With multiplex, we have more. Now, if it's duplex, it's 2. If it's triplex, it's 3 targets, quadruplex, 4 targets. So whatever, let's say, levels are available, and availability of multiplex actually depends on the capabilities of the systems to separate or, let's say, detect different fluorophores. And as mentioned before, 2 fluorescence channels were already available since the very beginning of digital PCR. Usually, decision for multiplex was connected to cost efficiency. However, let's say recently we are developing new assays that are designed actually to really enable new applications that were not even, let's say, imaginable before. But of course, the question is why transfer to digital PCR? One answer might be, let's say, inaccurate multiplex quantification when using qPCR. So we've been using a lot qPCR with multiplex as well, but mostly it was for qualitative purpose, so for detection, because there were quite so many problems with multiplex quantification. And of course, another factor of going into multiplex is the cost. So if you have a simplex reaction when you can quantify one target, or multiplex when you can quantify several, then of course the cost per reaction drops significantly. And let's say next factor as well, new applications, as I've mentioned. However, with multiplex assay design, there are a bit more challenges than just with the simplex. There can be interactions that are between the assays. Now you're putting more in your reaction, and of course, the level of interactions can be higher. There can be some non-specific amplification because you have more of those assays present, more interactions, and targets that you might not foresee can be amplified. The limits of quantification or detection might be higher. So there's quite some more work when you're going into the multiplex assay design, but in the end, it can be rewarding as well. So currently, the availability of multiplex digital PCR systems is actually better than ever. Here I have just an example of several platforms. Not all are listed here. Not all have been probably tested sufficiently, but just let's say what is on the market is, you can see from, let's say, 3 fluorescence channels to 7 with the newest Stila platform, so NIO Plus. And I did not go into comparison of all other parameters of those systems, but of course, if you're testing a lot of samples, then also the throughput is important. And when you already come to such parameters, there are perhaps only 1, 2, 3 of the systems that are still, let's say, viable to think about. So speaking about higher number of fluorescent channels also means that you can quantify more targets in the same reaction. So if you have more targets to amplify, that can mean the reduction of time that is needed to complete all the tests, and also in the improvement of price performance. And this is actually without having any compromises on the performance parameters, such as accuracy, precision, and repeatability. And as mentioned before, again, possibilities of new applications. I will show you now a case study from the gene therapy field. So about the construct delivery, as mentioned before, viruses are needed to deliver some constructs into the patients. And I will focus here on AAV vector genomes and AAV viruses. So the wild type AAV genome was actually modified in a way that it can transfer the payload, so the desired genetic construct, into the patient. However, when producing the recombinant AAVs, we can actually get pretty different content inside the vector capsids. So, of course, what we want is having the intact genome present in the capsids, but we can also get fragmented genomes encapsulated. We can have empty particles as well, or we even have the capsids that encapsulated impurities, such as, let's say, plasmid fragments or host cell DNA fragments. And until recently, the focus of characterization of those particles was on full and empty viruses. However, with development of new analytical approaches, the focus kind of also now shifted against partial particles, meaning those that are partially full. So by having, let's say, just some fragments of the vector genomes. However, when considering the full and empty particles, the methods that are kind of traditionally being used for that are not genome specific. So any, let's say, methods such as AUC, mass photometry, or TEM do give you some, let's say, peaks of populations, and then you can quantify from these peaks, but you never know what is included in these populations. And this is, let's say, still kind of a challenge. Then we come to the titering methods with the digital PCR. So as mentioned at the beginning, this kind of method was or already is a gold standard. However, there is one limitation of the titering methods that are currently being used, and this is actually that it targets just one specific part of the genome. And as it's exemplified here on this scheme, you can see that we actually have 2 full-length genomes and then some fragments. And if we do quantification on such sample, we get the results of 6. So we would have a viral titer of 6 copies. However, you can see that, of course, this is not the case. We can then target other parts of the genome as well with specific assays. And if we quantify with those assays, we are getting, let's say, similar results in a way, but still definitely it's overestimating the number of full vector genomes. And actually, to have a better insight into what is actually in the capsids, so that we are not surprised by any monsters hiding there, we need to put focus on the vector genome to get really genome-specific information. And vector genome integrity, meaning, let's say, characterizing the sequence or the construct as it is in the whole length, it's becoming quite an important quality attribute. There are increasing number of analytical approaches to characterize the content of the capsids. Also, number of publications for genome integrity is increasing, and also number of companies researching genome integrity is also increasing. And we kind of started with this genome integrity by developed, and we also qualified a quadruplex system for digital PCR to assess vector genome integrity. This was done within NIBA Labs, and we have this NIBAplex platform where we have 4 different assays positioned on the vector genomes. Currently, those that are, let's say, available off the shelf include those targets that are mentioned here. However, we have new targets in development for this kind of a mix and match approach, so that customers could combine different targets by their own, or let's say we even provide a custom design so that we can make these kind of assays completely customized. So what is now actually the good thing of this assay? We can actually follow what is happening with the process. We can compare the samples, so it has very good value in terms of comparability. And here we have, for instance, just an example of different samples that were being tested by this 4-plex assay to obtain the quantity of full-length vectors. And those were different assays coming from the optimization of the transfection. And as you can see, the minimum, let's say, and the maximum, what we've been measured in terms of the percentage of full-length vectors already kind of showed a 4-fold difference. However, we need to consider, of course, those that are marked here in gray that only have one small fragment included, because those could be purified out during the process. So these were harvest samples. And let's say during downstream purification, you could purify those out probably as empty particles. But kind of the additional benefit that we have is absolute quantification of different fragments that can be encapsulated. And if we just take a look at the full-length fragments between those 2 different samples, you can see that it's even 14-fold difference in the absolute copies of the full-length genomes, meaning that definitely we should consider this condition to optimize and go further with it in our manufacturing process. So if we just put now, let's say, our approach together with the scheme that we've previously seen, we can actually quantify, so not just kind of give some, let's say, approximate value, but we can quantify the actual content in terms of the different fragments of the vector genomes. So very, let's say, good insight into the population of viral capsids that we have. And kind of the key benefits of that are, of course, as I mentioned, you can inform the process optimization quite well. You have actually a better characterized product. And in the end, by moving into the producing more capsids that are full with your genome construct of interest, so with the full length, you could even lower the dose, so the production costs per dose could get lower. And with that, you could even dose the patients with a lower dose, because you would have more of the effective particles, and that would mean safety attributes to the patient. And of course, if you have an assay that gives you this kind of better insight, you can use it for comparability exercises as well, because any kind of, let's say, process modifications would probably also change this kind of fragment populations that you might have in the samples. So I hope that I've shown you really that this assay is applicable, and that the multiplexing kind of gives additional value. I can tell you that we have tested this assay on different platforms. It performs well. Stila's NeoPlus system was one of them. So we are now compiling all the results that we've got, but the results are very promising as well. And with that, I would then like to just conclude my presentation with some take-home messages, where the first one would be about the rate of digital PCR adoption, which we have seen it kind of increased really significantly through the last years. And as I said, it kind of really entered into this exponential phase. Then the performance characteristics of digital PCR actually make it a method of choice for really accurate absolute quantification. Then we have an increase in multiplexing capabilities that are actually even more interesting and useful. So I'm really curious what the future will bring in terms of those capabilities. So to really take the most out of the possibilities of having so many targets in one reaction. And in gene therapy, digital PCR is still a gold standard, and it will remain for accurate viral vector quantification. And I hope that it will also become a standard for new applications, such as higher multiplexing and vector genome integrity quantification. And as such, with, let's say, big adoption, I believe that more and more assays are expected to be run also in the QC labs using digital PCR in the future. And we are also working on development of assays that might be appropriate also for some impurity testing. And I believe that there are already a lot of labs that do use, let's say, impurity assays already in the digital PCR platform. So with that, I would like to conclude and acknowledge both of my employees for really valuable information that we got, and also to Stila for enabling this presentation. And with that, I think we can move to the Q&A session. Thank you.
Thank you very much, David. This was a really nice talk. Before we go into questions, I have one for you, which is, will you be present at any gene or cell and gene therapy conferences anytime soon where people may be able to meet you?
Yes, the first one would be in 2 weeks, ASGCT meeting in Baltimore. Then it will be Gene Therapy Analytical Development Europe in London, and Monolith Summer Symposium in Porto Roche in Slovenia, and beginning of June. That's okay. On the short term.
Okay, great. So we're going to be, also Stila will be at ASGCT in Baltimore in May, and in GTAD as well. So we'll both be at this event for everyone to know. So just to move on to the questions. One question that we have here is, do you purify the vector DNA before full-length characterization?
No. For the full-length characterization, we, of course, do the pre-treatment of sample to get rid of any let's say, free-floating DNAs. Mm-hmm. But then it is essential that we keep the vectors intact, because if we do release the DNAs, the vector genomes, we might introduce some additional fragmentation, so the results would not be as informative. But if we put those intact vectors in the reaction, then we can be sure that what we get in the reaction is actually what was already encapsulated.
Okay. That's very clear. Another question that we have here is, so sorry, it keeps skipping. Did you consider an AAV PCR system that was targeting the ITR regions of the AAV?
For vector genome integrity purpose, not. We have tried, let's say, with a kind of a proof of principle experiments when we kind of constructed artificial fragment with two such ends mimicking the ITR, just to see what kind of results we would get if we would employ this. Mm-hmm. But as you might imagine, if we have a signal from both ends, then the results would be, let's say, a bit vague, and we would not be able to really interpret them in a way as we can do it now. So, of course, we need to kind of skip the ITR regions, but we are trying to cover as much as possible of the vector genome.
Okay. And do you routinely titrate your samples in order to achieve the results in the linear dynamic range?
I don't know if the question refers to the...
I imagine, so I'm just reading the questions. I would imagine that the question is around the dynamic range. So do you need to dilute your samples to fit within the dynamic range of the digital PCR instrument you're using? I'm guessing.
Yeah, well, if it's just for digital PCR, as let's say in general, yes, you need to dilute the samples based on the, let's say, at least some expected concentration, because we might quickly saturate the reaction with such huge amount of targets. So that's perhaps, let's say, a disadvantage of digital PCR, that there is an upper limit of what we can put in the reaction. But yeah, with genome integrity, we need to be a bit even more careful because of possibilities of colocalization. So we need to consider that as well when preparing the reactions.
Yeah. Another question is around comparison with qPCR. So in qPCR, amplicons are advised to be between 50 base pairs to 200 base pairs in size. Would you say that there's a similar limitation in digital PCR?
Yes, usually it's quite similar. In terms of, let's say, going into longer fragments, digital PCR in theory could do that as well, but I don't have any experience by kind of amplifying longer ones. The efficiency of amplification would be lower and the fluorescence amplitude might be lower, so perhaps the separation of the positive negative clusters would be a bit more difficult. But let's say in theory it should work, but just for kind of, let's say, quantitative purposes, also for assays like genome integrity, we are using short amplicons.
All right. There's also another question about RNA vectors and whether you have any experience to share in the characterization of lentiviral vectors rather than AAV.
Yes, well, as I mentioned at the beginning, 2018, we already went into the lentiviral field as well by developing assays for quantification of lentiviral vectors. So it's actually no problem. But you need to consider that the vector is different, so the sample preparation is different. It's RNA vector, so you need to extract RNA and then use reverse transcription in one or two step approach. So that depends on the development, and yeah.
Okay, and maybe one last question, if that's all right. Let me just pick it. So another question also with regards to shifting assays from qPCR to digital PCR. So one person is asking whether you have any advice about how to do that and whether there are any usual issues that you might have moving your qPCR assays to digital PCR?
Yeah, well, definitely, let's say kind of the first step would be just taking the same kind of assay that you have with qPCR and test it on the digital PCR system. So let's say same concentrations of primers, probes, same amplification conditions. In some cases, you might see that, let's say, the digital PCR producers do recommend, or they have kind of their own recommended concentrations of primers and probes that kind of fit well to the polymerase they're using. So might be kind of good to consider this factor as well. But then, of course, if you don't get good results, you need to perform some optimizations, and there are many different things that can be optimized to kind of increase the signal or just improve the performance of the assay.
Okay, very clear. Thank you very much. Okay, so with this, we're going to close this webinar. Before closing it, just so everyone knows, if you've asked a question which we didn't have a chance to address, we'll be getting in touch with you by email after this webinar so that we can address it. So thank you very much, David, for this great presentation and for your time today. And thank you everyone for attending.